First of all, I'd like to thank the organizing committee for giving me an opportunity to present some results from my PhD project about arrhythmogenic right ventricular cardiomyopathy. So I'm going to talk about plakophilin-2 mutations in ARVC. So as we've heard from the previous talks, ARVC is a clinically and genetically heterogeneous condition. It is characterized by fibrofatty replacement of the myocardium, and these changes may involve both the right ventricle and the left ventricle. And the last decade's research have shown that approximately 40 to 50% of ARVC index patients carry mutations in genes encoding desmosomal proteins. And these desmosomes, they are very abundant in the heart, but also in other tissues. However, the genes are very polymorphic, and many mutations have been discovered so far. But unfortunately, little is known about the effects of the mutations, primarily because it's difficult to get cardiac tissue for protein studies. So in this study, we aim to establish a cell model system for studying desmosomal protein expression in ARVC patients carrying desmosomal mutations, and more specifically, we wanted to investigate effects of plakophilin-2 mutations on the gene and the protein expression levels. So we started out with our cohort of ARVC probands. We had 71 probands, and they all had a definite or borderline diagnosis of ARVC according to the recent task force criteria. And we sequenced the 5 desmosomal genes in these index patients, and we also invited the family members for clinical and genetic evaluation. So we performed primarily non-invasive investigations in the family members, and if genetic testing of the probands had identified a specific variant with a likely pathogenic effect, we performed genetic testing for this specific variant in family members. So our results were regarding to plakophilin-2 mutations that we identified 12 probands with mutations. And 10 of these probands, they carry mutations that were predicted to introduce a premature termination codon in the reading frame, so-called PTC mutations. We had 7 frame shifts, 2 nonsense, and 1 splice site mutation. Furthermore, we had 2 probands who carried missense mutations, and one of these mutations was a substitution of the first amino acid from a methionine to a leucine, and the other variant was a missense variant converting proline to threonine at position 103, and it is of unknown significance. Furthermore, we found that 3 of the 12 probands had a complex genotype, and they carried digenic variants. In the relatives, we examined these 12 families, and we identified 22 family members with mutations, but only 7 fulfilled recent task force criteria, and 5 had a borderline diagnosis of the condition, primarily because they had a pathogenic mutation and a normal number of ventricular ectopic beats or late potentials. And there were 10 non-penetrant carriers, of which 6 of them were had an age of 40 or more. So overall, the penetrance of plakophilin-2 mutations in our cohort was 60% if we included probands, but it was as low as 30% when we excluded the index patients. So in our studies, we wanted to investigate these mutations further, and one of our ARVC patients, he underwent cardiac transplantation. This gentleman, he had onset of symptoms since adolescence in terms of palpitations and dizziness, but no syncopal episodes. He was referred to us when he was 51 years old due to terminal right ventricular heart failure, and we listed him for transplantation. In the meantime, we performed genetic investigations, and He was shown to carry two mutations, a well-known stop mutation in the plakophilin-2 gene, in addition to a splice site mutation in desmoglein-2. A donor heart came available, and we got the explanted heart for further examination. This is his heart. First, we have the left ventricle here with normal cavity dimensions, but there's scar tissue here in the inferior part of the septum and the inferior wall. But his right ventricle is severely dilated, and almost all of the myocardium is replaced by fibrous and fatty tissue. We got a sample from the interventricular septum, and with kind help from Dr. Katle Gemlich in Oxford, we were able to investigate the expression of plakophilin-2 and desmoglein-2 in the heart of the patient. These are the Western blotting, and in the middle we have the patient, and the patient sample is flanked by control samples of two non-failing donor hearts. We use different antibodies with epitopes targeted against epitopes in both the N-terminal part of the protein and in the C-terminal part of the protein. And most importantly, we did not identify any truncated plakophilin-2 or desmoglein-2 proteins in this myocardium. And when we quantified, we found that both plakophilin-2 and desmoglein-2 expression was reduced by approximately 50%. So this indicates that the two mutations detected in this patient both lead to haploinsufficiency in the myocardium. However, it is only rarely possible to get myocardium for these investigations. So with inspiration, I forgot this. This is the ordinary immunohistochemistry of the myocardium, and this sample is stained with an plakophilin-2 antibody, and also indicates that there's less plakophilin-2 protein in this patient compared with a control. However, it is rarely possible to get myocardial tissue. So, with inspiration from the Naxos disease and Cavell syndrome, we wanted to look into the desmosomes in the epidermis instead. And all the cardiac isoforms of the desmosomal proteins are expressed in the skin, although in lower amounts. And this is a staining of a skin biopsy from this patient and a wild-type control. And as you can see, there's only very weak plakophilin-2 staining here in the basal epidermal layers. However, if we look into the epidermal glands, the sweat glands, we have a positive staining for plakophilin-2, and there appears to be less staining here in the mutant sample. However, when we do the biopsies, it's not always possible to get representation of sweat glands. So we wanted to set up another method. These glands and the epidermal structures, they all originate from the same stem cell, the primary keratinocyte. So we performed skin biopsies in mutation carriers and the wild-type family members. We took the samples here from the hip skin, and then we isolated the primary keratinocytes and cultured them in our lab in order to get sufficient amount for protein studies. This method is based on the paper written in 1975 by Reinhold and Green. So this is a picture of a confluent cell culture. These are primary keratinocytes, and we expose them to calcium in order to formate desmosomes, and this is a plakophilin-2 staining of the cells showing positive staining at the intercellular borders. This is the nuclei which have been stained by DAPI. So again, back to this family. This was the patient who underwent cardiac transplantation. He had no family history of ARVC. Both parents died from other causes. But we looked into his sons, and he had an 18-year-old son who was diagnosed with ARVC due to episodes of non-sustained ventricular tachycardia and fibrofatty replacement in biopsies, and he was shown to carry both mutations. An older son, he was healthy and carried none of the mutations. So this is a Western blotting of cultured keratinocytes from this family. We have the 2 mutation carriers here and the wild-type control in the middle. And there's lesser protein, plakophilin-2 protein present in the mutation carriers. So this corresponded very well with the findings we made in the explanted heart. So we extended our studies to skin biopsies from all index patients. This is a representative Western blot of what we found, containing different mutations from different index patients. And compared with wild-type controls, there's a generally lower plakophilin-2 expression in mutation carriers. And this is in absence of any truncated proteins. However, there was one exception. This was a patient with the missense variant in plakophilin-2, the proline to threonine conversion at position 103, who by quantification appeared to have normal protein levels. So we did repeated experiments, and this is the quantifications we did. We related the amount of plakophilin-2 to a loading control protein, beta-actin. And we observed that carriers of these PTC mutations had a significantly lower protein expression compared to wild type. And this was also the case in the patient carrying the missense variant in the first amino acid, but not with the other missense variant. So we also looked at the mRNA levels in cultured keratinocytes, and here we, in order to expand the number of samples, we included samples from family members who also carried mutations. And again, we see that there's a lower gene expression in carriers of PTC mutations. And that indicates that the mechanism behind lower protein expression in these patients is likely that the mutation leads to an unstable mRNA transcript, which is degraded by the nonsense-mediated mRNA decay mechanism. In carriers of missense mutations, we found normal gene expression levels. So finally, we wanted to reproduce the finding in the protein studies by another more accurate and antibody-independent method. So we applied liquid chromatography protein mass spectrometry in selected samples. And the advantage of this technique is that we can identify plakophilin-2 peptides with high accuracy, and then we can normalize the expression of the target protein with 10 other co-expressed proteins in the samples. So these experiments reproduced the finding from the Western blotting, and we found that the amount of plakophilin-2 was approximately 50% lower in carriers of the nonsense mutation, as well in carriers of the missense mutation at the first amino acid in the protein. And again, we observed that in 2 samples with the missense variant of unknown significance, there was not significantly reduced protein expression levels. So, ladies and gentlemen, based on these findings, I conclude that desmosomal protein expression in the myocardium is mirrored in keratinocytes of plakophilin-2 mutation carriers, and that makes keratinocytes a valuable tool in order to classify these desmosomal gene mutations in ARVC patients. And we found that most plakophilin-2 mutations, they are associated with haploinsufficiency. However, we also found that 50% of the relatives carrying these mutations, they were healthy. So that indicates that plakophilin-2 haploinsufficiency, when appearing in isolation, it appears to be well tolerated. So we think in order to develop ARVC, you need additional genetic factors present in order to develop the phenotype. So that indicates that the disease is caused by oligo or digenic inheritance of multiple genetic variants. At last, I'd like to thank my supervisors and my collaborators. Thank you for your attention.
I didn't understand well. All those who had reduced expression of plakophilin-2 were studied on the cardiac phenotype also. Did you make any correlation with the expression to the cardiac phenotype?
I mean, all the index patients
the
fulfilled.
individuals studied who had a reduced expression plakophilin-2 were relatives or probants?
Or you mean if you have, you wanted to correlate the expression of the protein with the clinical phenotype?
With the clinical cardiac phenotype?
No, we didn't have enough samples to do that, but
because the interesting thing would be if you can detect a preliminary expression from the skin without having the expression on the ECG or structural heart disease.
That's a good question, but we found that the PTC mutations, the frame shift nonsense and splice site mutations, they lead to approximately 50% in reduction in protein expression. And of course, there's an individual difference.
The biochemical phenotype of the skin, but what about the clinical phenotype in the heart?
As I said, in the mutation carriers and in the families, there were only a few affected individuals.
Yes, I mean, if you had a relation between the person who had this expression and the other, and what was the clinical expression in the heart?
No, we did not compare the clinical phenotype with the protein amounts.
It's the next step. Okay.
Sorry.
Thank you.
Okay. Yep, here and there.
Very nice data. I want to congratulate you for this because I think this is beautiful to be able to take the skin biopsy and to prove in the keratinocytes that you have normal expression of this wild type and mutant, and you can potentially correlate to the phenotype, but this will come later, I guess. So bravo. And I have a question related to this methionine.
Yes.
This is the first time I saw that the methionine is mutated, and I predict that you don't have any translation, or you have another methionine which is taken to make a bad protein folded. It looks like you have the same amount of mRNA in this patient, but there is a lower level of protein. So I guess you have no protein, no mutant protein.
Yes, that's also what we think, because the change of the methionine to the leucine is likely to inhibit initiation of translation. Yes. But we did not transfect cells with the construct, but that could be an idea to investigate that further. So, but in the plakophilin sequence, there could be an alternative start codon lower in the free end, but we did not identify additional proteins when we probed the bloods with both N-terminal and C-terminal antibodies. So I think this mutation also leads to haploinsufficiency, like the PTC mutations.
Yes.
Christina?
First of all, congratulations for a very beautiful work. And my question is, did you test plakoglobin also levels in this sample?
We did it some, but we did not found it to be reduced. And we also looked into our old myocardial biopsies, the formalin-fixed one. But we did not found any correlation between...
Yeah, because the original hypothesis, I mean, of the Safitz group was that the plakoglobin could be the final common pathway in terms of despite the mutation. So, and the second is, are you expanding your result to other gene mutation, I mean, desmoglein, desmocolin? Yes. What are you expecting from this investigation?
We've also looked into desmoplakin mutations and desmoglein-2 mutations, and papers are coming out hopefully soon. But with respect to desmoglein mutations, we found it to be another mechanism, rather a dominant negative mechanism of mutant proteins incorporated into desmosomes. And in desmoplakin mutations, it's more different because we both have missense variants and stop mutations. So there may be different effects. So that's why we suggest that this system should be used to classify these mutations. Because in order to develop good animal models or cell models, you need to know what is the fate of the protein in human cells of mutation carriers. So this is one of the strengths of this system, we think.
Okay. Is it absolutely necessary to do a biopsy? Could you not take skin scrapings instead?
Good question, but as you saw, there's very little plakophilin-2 protein in the skin because there's other isoforms of the protein. So that's why we wanted to culture the primary keratinocytes in order to get sufficient amounts for protein studies.
Yeah. Okay. Any other questions? Great. So thank you very much.